Very good morning to every one of you. Now, today we will have a lecture from Dr. Ashutosh from Motilal Nehru National Institute of Technology, Allahabad. There is some technical glitch or anything of that sort, network problem. He is not able to join. He'll be joining in a short while, and then we will start with today's talk.
Everyone, can you hear me, please?
Yes.
Am I audible?
Yes.
Okay.
Now you can share your presentation.
Right.
So we have Dr. Ashutosh Mani as our speaker of this session. He is an assistant professor at Department of Biotechnology, Motilal Nehru National Institute of Technology, Allahabad. He has done his graduation from DDU Gorakhpur University and his PG and PhD in Bioinformatics from University of Allahabad. He has been a member of several committees in MNNIT, and he has held the position of Member Secretary Institute Ethics Committee. He has been coordinator and treasurer of Kalrao, coordinator of NBA PG Biotechnology. He is convener of Departmental Doctoral Program Committee. He is laboratory in charge of biotechnology, and he is member of Departmental Faculty Advisory Committee. He has received several grants from SARB New Delhi. He has received grants from UGC. He has supervised many PhD theses and guided several MTech students. He has more than 30 research papers to his credit. Dr. Ashutosh Mani has filed several patents. He is reviewer of reputed journals including Nature's Scientific Report, and he has received many awards from CSIR and SARB. So now I would like to invite him to present his talk before you all, which is on the topic Role of Molecular Modeling in Development of Novel Therapeutics for Alzheimer's Disease. So now, Dr. Ashutosh Mani, please start with your lecture.
So, good morning, everyone. Let me know if I am audible. So, first of all, I would like to thank Dr. Nidhi Mishra for inviting me and giving me opportunity to interact with you all. And I am also thankful to her team for considering me as a speaker in this FDP program. So as it is clear, Dr. Nidhi Mishra told that today's talk is titled Role of Molecular Modeling in Development of Novel Therapeutics for Alzheimer's Disease. So from the first slide, we see here there is an introduction about the Alzheimer's disease. So before we proceed, I would like to discuss a bit about Alzheimer's disease. So basically, Alzheimer's disease is a neurodegenerative disease. It causes progressive impairment of cognitive functions. So it adversely affects memory, comprehension ability. It starts language problems, problems in paying attention, reasoning, and judgment. So this disease is basically characterized by accumulation of abnormal senile plaques and neurofibrillary tangles. What symptoms we observe in patients like loss of memory, language problems, difficulty in doing very simple tasks, disorientation, loss of reasoning capacity, difficulty in elaborating their thoughts. They start losing objects. There are swings in mood, or there are mood changes, and there are behavioral problems, and they lack initiative. They start do not start any new work or new job, or they have a problem with initiating any taking any initiative. So there are on the basis of genetics involved in progression of Alzheimer's disease or development of Alzheimer's disease, we can uh, divide them into basically three types of ADs or Alzheimer's diseases. First one is early onset Alzheimer's disease. So it starts early in early age, maybe in 30s, and it is due to genetic factors that are inherited, and it comprises only 1% of the total AD population. While there is another type which is early onset AD, that is caused by complex inheritance. So in this case, the disease start developing after 65 years, or somewhat around that age, and the cause behind development is genetic factors as well as environmental factors that may be linked to lifestyle or other environmental factors. So this type of patients comprise 4% of the total Alzheimer's populations. And the third one is late onset. It is also a complex inheritance type of disease, uh, and the causes are genetic as well as environmental factors. So it also develops around 65 years of age, and the population that is covered by this type of disease, or the factors, this type of factors in AD is about 95%. So majorly the Alzheimer's disease population is a result of a late onset AD, that is due to a combination of genetic as well as environmental factors. So they both together cause development and progression of Alzheimer's disease. Now we come to specific genes or proteins involved. So basically, uh, in early onset Alzheimer's disease, there are three genes that are identified and responsible for development of Alzheimer's disease. First one is amyloid precursor protein or APP. Second one is preslin 1 protein and preslin 2 protein. So if a parent is affected, child has 50% chance of inheriting the mutation, and presence of any mutation in these genes causes directly causes the disease, that is Alzheimer's disease. So now it is clear that Alzheimer's disease is a multifactorial disease, uh, having 70% genetic component and 30% environmental factor component. And among the genetic factors are genes associated with a family of the disease that are called familial Alzheimer's disease or FAD, or sporadic Alzheimer's disease or SAD. And the genes involved are APP, PSEN1, PSEN2, and above them, ApoE gene, amyloid protein E gene, is responsible for the sporadic form of the disease. So when we are talking about very large population, major chunk of population that is affected by this type of disease is due to this ApoE. that is responsible for development of SAD or sporadic Alzheimer's disease. Now we come to a very popular, theory, or most of the scientists around the world are involved in, exploring molecules or drugs that are targeting beta secretase enzymes or base 1 enzymes, and basically the theory behind targeting base 1 for curing or treatment of Alzheimer's relies on facts that amyloid beta peptides are generated by cleavage of amyloid precursor proteins, and they are cleaved by beta secretase or alpha secretase and gamma secretase. So when there is a mutation in amyloid precursor protein, so what happens? The cleavage site, is shifted to another site in the protein, and this causes production of somewhat, or some granules that are responsible for accumulation or aggregation of amyloid beta peptides, which ultimately form uh, you know which ultimately form some plaque type of things on the neurons, and that plaque disturbs the normal signaling of neurons, and that is why we observe very a lot of changes in the patients that are at neuronal level. So amyloid beta peptides are generally useful for neurons, but in Alzheimer's disease, excessive accumulation of intermediate amyloid beta peptide products or misfolded amyloid beta peptide products, amyloid beta dimers and oligomers start accumulating around the neurons. So they further aggregate to form protofibrils, fibrils, and finally it turns into senile plaques, brain senile plaques that are composed of proteinaceous deposits with beta sheet structure. So basically, beta site amyloid precursor protein cleavage enzyme 1, that is base 1, is responsible for formation of amyloid beta peptides by cleavage of amyloid precursor protein. Base 1 is a membrane-bound aspartyl protease, and it is a promising therapeutic target for Alzheimer's disease, because basically this enzyme is responsible for formation of amyloid beta peptides that ultimately form senile plaques. So in this image, we can see that a healthy brain, comparison between a healthy brain and mild as well as severe Alzheimer's disease brain. So here we see that cerebral cortex and hippocampus is quite healthy and their volume is too good. But after development of mild form of Alzheimer's, the cortical shrinkage can be observed, and there are also present some ventricles uh, that are still small in size, and we can also see that hippocampal region is also shrinking. At the latter stage, when Alzheimer's disease has turned into a severe form, we can see that cortical shrinkage has become more severe, and the ventricles have grown in size considerably, larger than the previous form, and as well as the hippocampal regions have also shrunk too much. So the overall the volume of the different parts of the brain starts shrinking due to Alzheimer's disease. here we can easily discuss, understand, as discussed in the previous slide, that what happens when amyloid uh, precursor protein is cleaved by gamma secretase and alpha secretase, that is non-amylogenic pathway. Non-amylogenic pathway means it does not generate amyloid beta peptides that are responsible for Alzheimer's disease, while amylogenic pathway is responsible for generation of amyloid beta peptides that form aggregates and create senile plaques, and which ultimately is responsible for development of Alzheimer's disease. So here we can see that this is in the center between non-amylogenic and amylogenic pathway. In the center, there is a protein that is amyloid precursor protein, and we can easily see the N terminal, the C terminal, and amyloid beta domain. So basically, the alpha or um, alpha secretase, that is alpha secretase, cuts, cleaves the protein in the amyloid beta domain. So a new protein or peptide is formed, that is having N terminal domain and a small part of the amyloid beta domain. And after that, the gamma secretase cuts between the amyloid beta part of the amyloid beta domain and C terminal, so P3 is released. So there is no generation of amyloid beta peptides. So it is non-amylogenic pathway, and it generally happens. It is a usual process in normal individuals. And on the right side, we see that there is beta secretase in place of alpha secretase. So what happens? Beta secretase cleaves the amyloid precursor protein at the junction of N terminal domain and amyloid beta domain. So after that, the gamma secretase, as usual, cuts the amyloid beta domain and C terminal domain directly between amyloid beta domain and C terminal domain. So as a result, P3 is not formed. Protein 3 is not formed while amyloid beta peptide is formed, and they are insoluble and capable of oligomerization. They cause neurofibril formation, and that ultimately results in senile plaques formation that disturbs the neuronal signaling, and we see or observe many behavioral and uh, new changes in the patients, like they start losing the objects, they forget the path through which they came, or they are not able to even recall very simple or latest things. So that is the basic reason behind the generation of amyloid beta peptides. So there is no it's obvious that anyone who will see or study this pathway will assume that this beta secretase must be inhibited to form some drugs that could bind on it and inhibit the formation of amyloid beta peptides. So many scientists are working on this. So as we discussed, I would like to summarize the previous image or figure. So
मला एक आपल्या लेटर हेडचा फोटो पाठव ना, नाही तर तुझ्याकडे ते वरती आपण असं टाकतो ते आहे का सॉफ्ट कॉपी मध्ये?
hello,
हॅलो,
is this? कोई कुछ कह रहा था क्या?
Sir, sorry for interruption. We have muted them all, the participants.
Okay, I'm sorry. I asked that. I thought that someone was asking some question. Okay, no
It
issue.
wasn't that.
So the precursor proteins are proteolytically cleaved by two distinct pathways, as I shown in the previous figure: the amylogenic pathway and non-amylogenic pathway. So amyloid precursor protein is cleaved within the amyloid beta domain by alpha secretase with the formation of a large soluble ectodomain, that is SAPPA, soluble amyloid precursor protein alpha, and an AT3 residue membrane associated C-terminal fragment, right? And in non-amylogenic pathway, amyloid precursor protein is cleaved at N-terminal of the amyloid beta domain by beta secretase or base 1. That results in the generation of a soluble ectodomain SAPPB and a 99 residue membrane retained C-terminal fragment. So that is a amylogenic pathway where beta secretase works. So the gist for some previous slides is that under normal physiological conditions, amyloid beta 40 is the most abundant species generated in amylogenic pathway. So that was a controlled measure where amylogenic pathway is always active even in normal individuals, and this amyloid beta 40 is useful for a healthy brain, and it is part of many physiological or molecular functions. But in this case, the amyloid beta 42 is only 10%. However, when there is a mutation in amyloid beta precursor protein APP, so the proportion of amyloid beta 42 is considerable, and they contribute to fibril formation, promote formation of amyloid beta aggregates, and they are key factors of neurotoxicity. So now we know that from where they come and what is the reason behind development of the disease. At least one theory we know now. So we can easily say, as the talk today is more oriented towards the drug designing purpose. So that is how from the literature study or from the previous findings we can identify the targets like beta secretase 1 in Alzheimer's disease for our drug designing. So I think this is from this figure we can say that they have shown which amino acids are prone to mutations, and which mutations are occurring at which place. So they have basically targeted the amyloid beta domain and acid acid domain or C-terminal domain of the amyloid precursor protein. And here we can say that K has been replaced by N, or M has been replaced by L at 670 and 671 residues. Next, we can see that A has been replaced by V, or D has been replaced by N. So, the most common amyloid precursor protein mutation changes the protein building blocks in the protein, and this mutation replaces amino acid valine with the isoleucine at 717. So this is the most common, most uh, important factor that is common in many patients. where valine has been replaced by isoleucine, and that is one of the main factors for development of or formation of uh, amylogenic, molecules in the brain. Now we come to another very important protein, that is presenilin 1 and 2. So basically, mutations in presenilin 1 gene that codes presenilin 1 protein formation are the most common cause of familial Alzheimer disease. So familial Alzheimer disease means it is due to genetic factors, and PS1 protein functions as the catalytic subunit of gamma secretase, and it means that somehow this mutation is also related with uh, cleavage of amyloid precursor protein by another enzyme that is part of both amylogenic and non-amylogenic pathway, that is gamma secretase, and they also take part in amyloid precursor protein cleavage. So following cleavage by the beta secretase, uh, where they account for formation of 90% of amyloid beta production, the minor amyloid beta 42 product is more hydrophobic and is thought to nucleate amyloid beta aggregation, leading to formation of plaques in the brain. So PSEN1 mutations initiate pathogenesis by increasing production of amyloid beta 42, because they also shift the cleavage site, and that results in formation of misfolded amyloid beta 42 peptides, and that contribute to the formation of, again, that contributes to the formation of uh, non-soluble or toxic amyloid beta peptides, and they form aggregates and contribute to the senile plaque formation. Now we come to the third type of Alzheimer's disease, that is late onset Alzheimer's disease. So basically, the many genes have been identified that are mutated in late onset Alzheimer's disease, one of the most common types of Alzheimer's disease. But apolipoprotein E, ApoE, is one of the major factors. It is considered as one of the major factors for development of Alzheimer's disease. So basically, apolipoprotein E is a protein involved in the metabolism of fats in the body, and it belongs to a family of fat binding proteins that is called apolipoproteins. So its length is 299 amino acids, and it has N-terminal from residue 1 to 167, and C-terminal is residue from 206 to 299. So it is a mixed type of protein that contains hydrophobic as well as hydrophilic domains within the same protein that are connected through hydrogen bonds, and they have their C-terminal domains have capability to bind with low density lipoprotein receptors, or LDLR. So basically, the um, hypothesis of considering ApoE being responsible for generation of AD is that there are four types of ApoE proteins: ApoE, ApoE2, 3, and 4. So It is considered that ApoE4, E, E2, and E3, they are involved in uh, clearing off the amyloid precursor proteins parts or the cleavage products uh, from the neurons or the from the uh, cellular part, but somehow where ApoE4 is more active or produced in larger amount, they are the clearance is hindered, or the clearance does not go at its usual rate, and that causes problem because when the amyloid beta peptides are not cleared off from the neurons, they start accumulation, and they develop toxicity, and as a result many complications start occurring which contribute to Alzheimer's disease. So basically, there are several sites in the amyloid, ApoE4 proteins, and that are prone to mutations, and some of these mutations contribute to change functions, and which ultimately adversely affect the normal functioning of proteins. So. Uh, Alzheimer's disease is characterized by buildups of aggregation of amyloid beta peptide. So, amyloid E protein enhances proteolytic breakdown of this peptide, both within and between the cells. So, ApoE4 is not as effective as others as in clearing off these, proteolytic breakdowns uh, products, and they result in increased vulnerability to Alzheimer's disease in individuals with that gene variation. Now we can summarize this part of my presentation with the help of this figure, that when there is a cleavage of amyloid precursor protein by alpha secretase, then they form the growth factors or normal peptides that contribute to physiology and molecular biology in a normal way, and that is a non-amylogenic pathway. But when beta and gamma secretases cut the amyloid precursor proteins, then amyloid beta 40 and amyloid beta 42 are formed. So basically, amyloid beta 42 start aggregation, they form oligomers, they form amyloid plaques, and they are neurotoxic, and they are responsible for neuronal cell death. So amyloid beta plaques as well as oligomers both are toxic, and they are responsible for neuronal cell death. So when neurons will start dying, they will it will disturb the signaling process, and we observe the obvious changes in the patient's behavior. And apart from this pathway, there is as we discussed, that ApoE4 is also involved, that is responsible for clearance of the these toxic peptide products. But when ApoE4 is hyperactive, they are the rest of the ApoE proteins are not that much active, and it is also responsible for accumulation of toxic protein products or toxic peptide products, and the disease is becomes more severe or in later state. And from the oligomers that are formed after aggregation of amyloid beta peptides, we see that they also disturb the kinase activity of kinases, and these are the proteins or enzymes that are responsible for many post-translational modifications in the proteins, and this ultimately adversely affects the calcium signaling cascades, and they start formation of tau tangles, tau precursor proteins, and they, like amyloid beta, amyloid plaques, there is also formation of tau tangles or tau filaments. So they together form neurofibrillary tangles. So neurofibrillary tangles are also responsible for neuronal cell death. So from this figure, we can get an holistic approach that how even a single, uh, you know, uh, hello, how a single change at or mutation in a crucial protein can affect two or three different pathways that can contribute to the generation of Alzheimer's disease, development of Alzheimer's disease, and that is how we can target, we can get a clue that what shall be our basic, prime strategy to identify a drug target or therapeutic target, and by inhibition of that target, we can be able to treat or cure the disease, or at least minimize the speed of development of the disease. So that is very important for drug designing purpose. So there are now some of, you know, it is a list of the drugs that are given to the patients who are suffering from Alzheimer's disease. So donepezil, rivastigmine, galantamine, memantine, donepezil and memantamine combination. So basically, all these drugs are cholinesterase inhibitors. So they are inhibitors of cholinesterase, and that binds to this enzyme that is responsible for hydrolysis of acetylcholine. So we all know that acetylcholine is a neurotransmitter, and cholinesterase is the enzyme that degrades acetylcholine. So what happens when we give an cholinesterase inhibitor to the patient? It binds on the cholinesterase enzyme. So the uh, acetylcholine that is present in the brain is not degraded easily, or it persists in the brain for a longer time. As a result, the normal, neuronal, the patient is able to sense or get the signals for a longer time, and normal neuro coordination is observed, and we can see that some symptoms are some physiological functions in patients have improved. That is long lasting, or some we have enhanced the life of neurotransmitters like acetylcholine present in the brain. As a result, neurocognition is improved in patient. But the problem is that it is it just treats the symptoms. It is they Most of these drugs are not able to cure the disease, because they are just treating the symptom. They are trying to keep the acetylcholine for a longer time in the brain, and that ultimately improves the neurocognition. But as the patient stops taking the medicine, so the symptoms of the disease also again prevail. So there is always a need to enhance the efficacy of the present drugs, or design or develop such drugs that are uh, uh, able to target a different molecule that is more uh, key feature in development of the disease. So based on these findings and literature, and the need to develop new drugs or new therapeutic targets, we uh, compared the gene expression profiles of 14 brain samples from different patients, and we had taken the data from EMBL, DDBJ, and Array Express, and our gene expression analysis uh, resulted in shortlisting of 5 genes that are highly upregulated, and their p values were also significant. So it was surprising for us to know that all these 5 molecules are not having any direct correlation with the amyloidogenic pathway, or amyloid precursor protein, or ApoE4 protein, or any other enzyme or molecule involved directly, that we discussed in the previous slides. But still, we explored the types of these molecules, their functions, and we found that basically all these shortlisted molecules are uh, fall under a category that is type of proteins. They are called G protein coupled receptors. So it was surprising for us. They are all G protein coupled receptors, and that's why we were interested in these GPCRs. So I think we all know that what are GPCRs. G protein coupled receptors are membrane bound proteins, and it is clear that their one end is inside the cell and one end is outside the cell, and they are characterized by presence of 7 transmembrane helices, and they are connected to extracellular loops. And one thing that is very popular or common in GPCRs is that they are very hard to crystallize. So basically, their crystallization is a very tough job, and that's why many bioinformaticians are involved in prediction of structure of GPCRs. So if we have the correct homologue present in the database, we can predict the structure of these GPCRs. So now we come to one by one at the shortlisted genes. So CHRM5. So CHRM5 is a cholinergic receptor, and as I said that all these are GPCRs, and it is responsible for binding with acetylcholine, and it is very much useful in many neurological functions. And acetylcholine is, as I told in the 2 or 3 slides back, that it is very important for Alzheimer's disease patients. And the why this CHRM5 level were increased in Alzheimer's patient was not is not clear, but you know it is supposed that if a receptor is highly expressed, then it shall be readily able to bind with its natural ligand. So if it is receptor for acetylcholine CHRM5, then it should bind with acetylcholine, and normal signaling should occur. So brain shall be healthy, but somehow it is upregulated or highly expressed, and still the brain functions are lost in patients. Now we come to Cis Ltr2. So Cis Ltr2 is a cystenyl leukotriene receptor 2, and it is involved in many diseases like melanoma and asthma, and gene ontology and notations have related this gene to GPCR activity and cis-LTR activity, and its important paralog is cis-LTR1. Next one is DDR5. So I think everyone has studied or read something about dopamine. So dopamine is responsible for good mood and for high spirits, and we all know that dopamine is known to make your mood good. So you feel well if the dopamine is secreted. So dopamine receptors, are also enhanced. Their expression is enhanced in uh, Alzheimer's disease patients. And STR2C again is a receptor for serotonin. So serotonin is basically for uh, patients that are having serotonin should have good appetite and digestion, but somehow it is not observed in Alzheimer's disease patient. So it seems that the STR2C is also overexpressed in Alzheimer's disease patients, but somehow its natural ligands are not present. And the last molecule that was shortlisted is GAL-R1. It is also a GPCR protein that binds with galanin. So galanin is basically responsible for waking up, sleep regulation, cognition, feeding, regulation of mood, and regulation of blood pressure. So if galanin is also disturbed in someone, so these processes like walking, sleep regulation uh, shall be affected, adversely affected. So now we come to the drug designing part of our talk, where from these findings we thought that we shall identify some inhibitors that could bind on these overexpressed uh, molecules or receptors. So there is a very small introduction about different branches of bioinformatics. So we have a very one part of bioinformatics is dedicated to biological databases, then where these are repositories for the sequences for structures for structures of biomolecules, and they may also contain various miscellaneous databases, as we discussed that gene expression data may also be present in biological databases, and from where we can download our experiments results and we can compare them, so that we could identify overexpressed genes or underexpressed genes and many other things. So it helps us in finding appropriate places from where we can get the authentic data for our experiments or study. After that, we come to sequence analysis that includes pairwise comparison of sequences, multiple sequence alignments, and comparative genomics. So comparative genomics has many sub-segments or subsections. So expression analysis or data expression analysis, NGS data expression analysis is also a very advanced and recent branch of this comparative bioinformatics through genomics, and we are able to identify many genes that are upregulated or downregulated in diseased individuals, and or from the experimental data, and we can target some proteins or some genes as per our convenience. So example is the previous slide, where we had taken the data from Array Express, and we analyzed this next generation sequencing data, and ultimately we ended up in finding some genes that are over expressed or upregulated in Alzheimer's disease patients. So other types of other branches include molecular phylogeny, where we determine or uh, compare the phylogenetic information, we construct phylogenetic trees, we establish phylogenetic relationships among different organisms or sequences or genes. And very important for drug designing is structural bioinformatics. So in structural bioinformatics, we perform protein structure prediction, we perform molecular docking, we perform we perform drug designing, and we perform molecular dynamic simulations to study the molecular behavior at atomic levels. That includes the assessment of stability of a docking complex or a protein or any other biomolecule under a given environmental temperature, pressure, and other conditions like salt stress. So basically, when we are talking about drug designing, basically we are dealing with structural bioinformatics, but its root are also somewhere in databases and sequence analysis as well. So they are all co-linked, and they are interrelated. And basically, there is another field that is very important is development of biological databases and softwares. So that's a very small introduction about different branches of bioinformatics. So in this slide, we can see that we have sequence databases. We have like NCBI, GenBank, DDBJ, and EMBL, and we have structural databases from the protein data bank. So if someone has identified a target like this, it is easy that to explore that whether these structures are present in the protein data bank or not. You can just enter the name of the protein and organism in protein data bank, and you can know you will come to know whether this structure has been determined by X-ray crystallography or NMR, or it is not present. So if it is not present, then you will have to use this protein structure prediction strategy for knowing the three-dimensional structure of your protein, target protein. So now we come to the workflow of structure-based drug designing. So there are different starting points, but if you have a target enzyme or a receptor, as it is in our case, we can determine the three-dimensional structure with the help of X-ray crystallography, NMR, or electron microscopy. That is nowadays cryo-electron microscopy is very popular, and homology modeling. So if previous structures are not available through the previous experiments, we can also perform homology modeling for our target or receptor, and uh, we can screen suitable compound from different online libraries, or we can create our own library and we can screen them for our for binding with our receptor or enzyme, and we perform docking so that it forms a receptor ligand complex, and then after we can do other steps. We can go through other steps like QSAR, or we can if there is no proper binding between the receptor and ligand, and we think that it is not stable enough. Suppose MD simulations have suggested that the complex is not stable, then we can redesign for its affinity, and we can redesign the ligand and see whether it is now the binding score has improved or not, and we can optimize the overall molecule for better compatibility with our receptor. So once it is established that they are forming a stable complex, we can go for various pharmacokinetic testings in vivo as well as in silico approaches, by following in vivo as well as in silico approaches, and in many cases we see that most of the ligands that we have screened do not follow the Lipinski's rule or QSAR models, or they are not at par with the ADMET parameters. But still, in the at the end, in the last, we are able to shortlist a few molecules that are fitting in these parameters, and we say them that they are lead compounds, or we can go ahead with them for in vitro as well as in vivo testing. So overall, computer-aided structure-based drug designing has minimized the time required for finding a lead molecule, or a molecule that is having promising properties to inhibit a target or receptor. So it is very interesting, in fact. So suppose our molecule is not determined, its structure is not determined previously, and we have certain uh, ways to predict the structure. So ab initio means from beginning, from scratch, and it is followed when the sequence similarity between the template and the target is in the range of 0 to 20%. Fold recognition is generally used when the identity between the target and the template is 20 to 35%, and maybe in some cases, if you are targeting very good quality structure, it may be 40% also. And homology modeling is considered appropriate when the similarity between the target and the template is greater than 35%. So, for especially for drug designing, the models produced after homology modeling are considered suitable. So, for because the problem is that most of the models generated through ab initio modeling or fold recognition are not of appropriate quality. So you will find that many amino acids fall in unfavored regions, and there are they make a larger fraction of overall amino acids present in that protein. So basically, most of the molecules shall be in favorable regions in Raman of good quality. So how we identify a target? We identify a target from differential gene expression expression studies like 2D electrophoresis, real-time PCR, microarray, subtractive genomics, and now next generation sequencing is very popular. And these techniques have provide an initial idea about the targets. Mechanism-based studies and pathway analysis are required for confirmation of the targets. So, where we get the structure, so if the structure is present in protein data bank, and so it has solved our problem. We need not to again predict the structure of this protein, and if it is not present in the protein data bank, then obviously we have to look for a template that is present in the protein data bank. Template is a molecule whose structure has been determined by X-ray or NMR, and uh, our query protein or receptor protein is very much similar to that template protein. So that template may be chosen for homology modeling, and in some cases when where we do not find the one template that covers the entire stretch of the query protein, in that case we can also perform multi-template homology modeling. So it may also be needed for some protein. So what we did in this uh, study, we had taken the 5 receptors, and it was obvious that uh, all of them were G protein coupled receptors, GPCRs, and they are hard. They are not easy to be determined by X-ray or NMR. So their structures were not present in the protein data bank. So we used homology modeling technique to predict the structures of these proteins. So here in the table we have a list of used sequences with their accession numbers, amino acid lengths, and the used templates for the 3D structure prediction of the 5 GPCRs. So in the table number 2, we see that we have at the serial number 1 CHRM5, Cis Ltr2 at the second number, and DDR1, STR2C, and GAL-R1. And in the last column of the table number 2, we see that the templates that were used for the homology modeling. So we can see here that for each protein we have used 4 or 5 or sometimes 5 or 6 templates. So it means that we have performed multi-template modeling, because any single template was not covering the whole stretch of entire length of the our any of our proteins. And in the table number 3, we see the Ramachandran plot statistics that was generated by ProCheck validation server for all the 5 receptors. And we see here that in most of the cases, the uh, the most favored regions or allowed regions especially have more than 95 or 90% residues, and very few residues are were present in non-allowed regions or disallowed regions, and all these residues were removed from the disallowed regions by loop modeling. So loop modeling is a method where we you know re you know re design or reallocate the three dimension of the atoms involved in that particular residue, and so that they adjust themselves in a more comfortable state. So if any residue is in disallowed region, it means that the psi and phi angles are not at par with the Ramachandran plot, and they shall be adjusted in a relatively less stressed state. So it allows them to adjust themselves accordingly. And after loop modeling, generally the disallowed regions are shifted to allowed regions, and that enhances the overall number of residues falling in the most favored regions. So, as I told that the phi and psi angles were calculated to estimate the quality of the predicted structure for all the five uh, proteins, and most of the residues involved in constitution of these proteins were present in allowed regions, and overall quality of the structures was found to be good. So this is an image for Ramachandran plot of the five receptors that were predicted by homology modeling, and very few residues are present in disallowed regions, and they were removed after loop modeling. Now, after the this structure prediction, we have to perform docking. So we had chosen natural compound. We had created our own library of natural compounds, and we performed docking with the help of Glide, and we found that we shortlisted top two natural compounds that are binding with each of these residues, and we have mentioned the zinc IDs of the top two binding molecules with each uh, protein taken in the consideration, and basically we had shortlisted the overall docking score as well as glide score. And this table number 5, shows top 5 best protein ligand complexes showing the highest binding affinity between protein compounds and along with their interacting residues. So in this table, the column number 1 and 2 are same as taken in the previous table, as shown in the previous table, but the interact in the column number 3 we have shown the interacting residues, the residues from the protein CHRM that interact with this gene CO4098704. Okay, so in the same way we have shown the interacting residues, and this is the image that shows the interaction between the uh, receptor and the two most high scoring ligands that are binding with these receptors. So this is for CHRM5, Cis Ltr2, and DRD5 and GALR1, and 5st2c. So that's how, and at the bottom we can see that which colors or which colors lines or which structures show what type of interaction is there between the ligand and the receptor, or even between the atoms, right? So finally we have shown that what are the docking scores. So I think we have discussed this slide earlier in the previous slide. And these are the structures of the natural ligands that are binding with CHRM5. And we have mentioned the zinc numbers. So zinc is a database from where you can uh, screen a suitable ligand for binding with your receptor or target molecule. And after you are convinced that these are safe enough, efficient enough, effective enough to bind with your receptor, and they shall be safe enough also. They should follow Lipinski's rule. They should follow ADMET parameters, and then after we can proceed for in vivo as well as in vitro studies, in vitro as well as in vivo studies. So in the last, I just wanted to again discuss a very small, uh, topic, that is what are, we shall also always have a thought of an alternative, approach for the existing uh, study, because all the findings are just a result of experiments, and there are always some assumptions involved, and we may not be knowing everything every time. So I had just thought that why I shall not think that uh, uh, if everything is fine in GPCRs, they are highly, uh, expressed in Alzheimer's disease patients. So one thing is good, that I can design inhibitors for anything that is overexpressed, that is possibly causing a disease, and any, their suppression will find, ultimately or finally suppress the symptoms that are resulting in disease, or at least the patient will get some relief from the symptoms. But we shall also have an alternative approach for each finding. And I thought that if everything is fine, then why these GPCRs shall overexpress themselves? What may be the possible regions? Because if GPCRs are overexpressed, they are meant for normal or smooth, neuronal signaling. So why they are if they are available for binding with their natural ligand, if they are available for binding with their natural ligand, then normally the patient shall not suffer. They should any adverse symptoms shall not be visible, or they shall not be expressed. But uh, and in order to find an answer for this query or question, I thought I surveyed some literature. I explored the data, and I found that very in 10 or 20 10 or 12 years back, in a paper, uh, it was written that G protein couple receptors are very prone to an inverse agonism. So inverse agonism is a phenomena in which any molecule binds on the receptor, any molecule that is not natural ligand to that receptor binds on that receptor, and the normaling, and it brings it in an inactive form. So normal signaling or further cascade is inhibited. As a result, the molecule is expressing itself. The receptor is expressing itself, and each time it is being expressed, it is being synthesized in the cell. Some molecule that is non-specific for that receptor is readily able to bind that receptor and arrests it in an inactive form. So this forms an inverse agonism loop, and this is an open question that which molecules are causing the this type of inverse agonism loop formation, because at the time of amyloid precursor protein cleavage and in the normal physiological functions also, many molecules would have been generated, and if this new approach is given proper attention, we may able to find the exact reason behind the overexpression of these normal, these GPCRs that are responsible for neural, uh, signaling, neuronal signaling. And if they are overexpressed, then neuronal signaling shall be more smoother, and but it is not the case. The patient is still suffering. It means that something is bind on this transmitter, these receptors, that is causing them to be arrested in an inactive form. So that is just an hypothesis, and I think I have done with my slides, and so that's all for today's talk. Thank you very much. Thank you.
Thank you, Dr. Ashutosh Mani. We have two questions from Richa. She asks that is there any role of beta secretase in metabolism other than in neuron?
I'm unable to hear you, Dr. Nidhi.
Sir, she asks, is there any role of beta secretase in metabolism other than in neurons?
No.
Okay. And then she asks, a few literature says that amyloid beta could travel via the vagus. How justifiable it is to recognize a peptide traveling through nerves?
So, how it is justified to recognize a peptide? Actually, what I understand that we are not interested in identifying that peptide, because it is a normal function of ApoE proteins to clear off such insoluble protein products, and they are readily identified by ApoE proteins. And in rare cases where ApoE4 protein production is overexpressed, and there are some mutations in other proteins, as a result, they are not able to clear off the undesired protein products like amyloid beta 42 protein fragments. This causes increased toxicity in neurons, and further cell signaling is also adversely affected. So basically, so far there is no approach for targeting the trafficking of these peptides or base one products. Basically, they, as I told in the previous slides also, that in normal people also who are not suffering from this disease, these peptides are formed, and but the case is different in other individuals. In AD individuals, is that the expression or the production of such non-insoluble or toxic peptides is enhanced due to some genetic or maybe some environmental factors. So there is still a lot of debate that what is the exact reason behind generation of the disease, because whatever we know, it is from the experiments, and what lifestyle is exactly responsible is still a matter of debate. But what we have talked in this session is all about the previous findings and prevailing hypotheses. Hello, can you hear me?
So she asks about subtractive genomics.
Okay, so in subtractive genomics, we compare the genomes, or two genomes, or in some cases many genomes also, and we there is a host like human, or there is a pathogen, maybe a fungus or any other parasite. So the prerequisite is that the genomes of both organisms shall be known, and we compare them so that we could identify those genes that are exclusively present in the pathogen, and targeting those genes that are exclusively present in the pathogen will pave way for designing safer molecules, safer drug molecules. Because if a molecule, if a gene is not present in the host, it is present in only in the pathogen, then obviously it will there will be no non-specific binding with the host's protein. And if there will not be any non-specific binding with the host protein, so it will not harm the host, that is human. It will only target the pathogen. So subtractive genomics is very useful in that way.
Okay, so Sarab, there are no more questions now. Then somebody asks about multi-template modeling.
So multi-template modeling is possible through modular. I think most of you will be using modular, and it is a very handy, easy to use tool for protein structure prediction. So what we have seen is that generally, if there is a template or a similarity, it is over all the stretch of the from starting to the beginning in the whole stretch of the protein. But in some cases, we see that we have a query sequence, and we found a template whose structure is known, and it is present in the protein data bank. But say that our sequence is 100 amino acids long, but the template is covering only uh, starting 40 amino acids, say 1 to 40, though the similarity is say 90% or 80%. But the problem is that for our amino acids that are in the range of 60 to 100, we do not have any template. So in absence of a template for that stretch, we will not be able to predict a good structure for that region. So when we will analyze our result in Ramachandra plot, we will see that the quality is not good. So we do multi-template modeling, where we again explore, we with the help of blast search, we not only see the first hit, we also see the second, third, and fourth hit. So what happens? Maybe a second hit is there, or third hit is there, that is able to cover 60 to 100 amino acids that are tail portion of our protein. And maybe the similarity is not 80%, not 70%, but maybe the similarity is 50% or 40%, but still it is good enough to provide to work as a template rather than proceeding ahead without a template. So that is called multi-template modeling.
Thank you, sir, for explaining it. And there are no more questions in the chat box. So now
Okay.
I thank you, Dr. Ashutosh Mani, for such a wonderful talk and for clearing all the doubts in the minds of participants. I thank you once again on behalf of organizing committee and participants both. Thank you so much for coming to this event and sharing your thoughts.
So thank you, Dr. Nidhi Mishra and your whole team and Triple IT Allahabad for giving me opportunity to interact with the participants of this FDP, and it was really pleasure for me to interact with you all and share my experiments and results with you all. Thank you very much.